Hello, everyone. Welcome to AP Bio. I am so excited to be back here on a wonderful Monday. I know there's been a lot of upheaval, a lot of crazy changes going on. And so if you weren't here with us last week, we are doing these sessions and we're going to cover some pretty awesome questions together. And it's not going to be a comprehensive review on these topics, but we're going to kind of go through some of the skills and strategies necessary to be able to tackle some questions. In the topic for today will be cell communication and enzymes. So as Nick mentioned, please make sure you've got the little live button if you are not hearing this right now, then probably you aren't hitting it live. But yeah, make sure that little button is glowing red, and that way you're watching this live and not just a recording of it. Okay, so let's get started on this. Okay, so throw me a yes or a hi or a purple when you see the shared screen saying AP Biology, Cell Communication, and Enzymes. Let me know that you can see what I'm seeing so that we are all on the same page together. It's good times. Up here in wonderful Canada, it starts snowing again because, you know, it really wasn't enough that we got a pandemic going on. It had to start snowing. So we said goodbye to spring. We've headed backwards in time into winter. It's good times. Okay, so cell communication, enzymes, purple. Flora says purple. I'm glad that somebody else sees it. Okay. As always, I am joined by our fabulous off-camera instructor, Nick. So if you have any questions, if you want to chime in, anything, honestly, this is your session. This is for you guys. I don't really want to just sit on camera and blab. So I really want you guys to engage and interact with us as much as you can. Hello. I'm glad to see you all back here. Okay, so what I thought I'd do is, so the objectives here, we're just going to do some free response questions today. We're not going to do any multiple choice. Try to really work towards getting as much information from the College Board with regards to AP tests. Now, we don't know any more than you do. We don't. The official information will be coming out April 3rd. And once we have some more information, definitely keep on track of that. But what we're going to do is tackle some free response questions as dictated by the College Board prior to all of the crazy changes that have been happening. So we're going to do that in the topic of cell communication and enzymes. But I'm going to tackle free response questions in the style that was presented for, the current test. So that would be like experimental analysis, that would be drawing conclusions from data. It's going to be about proposing experiments. It's going to be about a little bit of graphing. So really the big idea of like, are you a scientist? Can you be a scientist? Can you put yourself in the scientist's shoes? And so Nicole, as of this time, we don't know any more than that. It will only be 45 minutes long, and it will only be free response questions. So if you kind of follow that logic, probably no grading questions, no multiple choice questions. Yeah, biology is biology. I completely agree. So if you know the content, you just want to make sure that you're practicing the skills, like how is this going to be tested? So yeah, I thought before we'd head over onto the very first question, before we do that, we just like really quickly, here are some tips for some free response questions. And I would make sure to get your hands on as many practice questions as you can, because once you have the content down, it's about the application of knowledge rather than just the recall, especially if the free response questions are going to be like they were dictated by the College Board earlier. So you only get points for the information that you get. You don't get any points for essentially filling in space. You know how you guys do that when, you've got an essay done and you're trying to like fill up the word count? Don't do that. Don't do that. All it's going to do is like steal time away. And since we don't know how many questions are going to be and how many parts to every question, it's always going to be a good, wise decision not to spend more time than you absolutely need to. So you don't really get anything more for just adding the fluff, okay? Don't go in circles, right? And this is kind of easy to do, especially when you feel like you're in the moment of like, I gotta fill something in here. If you contradict yourself, if you got it right at the beginning and then you like go in circles and then you contradict yourself later on, you're gonna get that point taken away, even though you had to state it correctly earlier. So don't do that. Don't go in circles. Grab the easy points. State the obvious, like, what is an acidic pH? We're going to see that a little bit. And then, provide a brief, accurate explanation. No need to go into a ton of content. And then the next one is don't use bullet points. So don't like outline things. Don't do anything like that. Have complete sentences. This is not a grammar test, but correct spelling will help the person reading it follow along a lot better. So use complete, and they don't have to be like fancy, giant, long sentences. If they're short, that's fine, as long as the information there is correct. Start with the questions that you're most comfortable with. And I cannot stress how important this is. You are not bound. You are not bound by doing the questions in the order that they're presented to you. Look for the easy ones first. If you are in a situation where you run out of time, you don't want to run out of time on the questions that are easy, right? You want to get the points you can, and then with the time remaining, tackle those tougher ones later on, okay? Base your response on facts, not feelings. Probably easier to do in the sciences, maybe less so in some of the more opinion-based kind of things, but base it on the facts. If you're given data, base it on the data. If you're given, asked about a scientific concept, base it on your recall of that content. Don't feel like you have to persuade somebody. This is all about the cold, hard facts. And then finally, if you use a term in your explanation, make sure you define it. So, talking about osmosis or hyperosmolarity, just mentioning that word might not get you the point, but if you define what hyperosmolarity is, then it is more likely that you'll get the point associated with that part of the question. So make sure if you use a science term, you take a second and you define it in your explanation as well, okay? So who is ready to do some questions? So previously, I always, in previous sessions, we did end the last one with like some possible kind of free response. I'd like to give you a little bit more leeway into doing this as well. So I'm going to give you what I think would be a reasonable amount of time. Now, I'm gonna be honest, I'm not 100% sure how much time you should be devoting for, what I've constructed in these questions. And so if it turns out that you're like, wait, it's too fast, yeah, I need more time, that's okay, just let me know, okay? So what I would like for you to do is take 3 minutes for this prompt. It's called a stimulus, we call it. So 3 minutes for the stimulus and the one associated question that's on the slide. And then I want you to grab some paper, and then write out your answer. Try to make sure that it is complete sentences with the correct spelling, defining your terms, things like that. And then we will compare and contrast notes. I will talk about how to approach it, what are some key things to keep an eye out for, and then we'll, lather, rinse, and repeat, okay? So here is your very first question. You can see some data, so make sure that you are, analyzing the data correctly. I'm going to give you 3 minutes to this one on your own. When we come back, we'll talk about it together. Okay, so tell me, was 3 minutes enough time to analyze the information in the stimulus and then to devise a possible experiment that would test this researcher's hypothesis? I feel like it's on the border. If you're really on top of knowing how to analyze data and knowing, what would be components of constructing an experiment, 3 minutes would be about enough time, but it is pushing it a little bit, especially if you're having to like write down a lot. So if you felt like that was a little bit tight on time, I think I'd probably agree with you. And so maybe about 4 minutes might be a more appropriate amount of time for a question like this one. But in any case, we are going to move forward with the review here. What I'd like for you to kind of think about is the information you gain from the stimulus prior to you trying to devise an experiment. You need the context first in order to kind of tackle, put yourself in the researcher's shoes. So the background information, we've got C. difficile, and it secretes a toxin. And so that toxin then does extensive tissue damage. They're trying to figure out, the not the mechanism, but like more information about that secretion by measuring how much of that toxin accumulates in the broth in which the bacteria grows in. Now, this is a part you really want to pay attention to. When you are given hypothesis statements, very likely that it's going to be a little hard. It's going to be a little hard. It's going to be really, really important. So pay attention to hypothesis statements. Nicole, that's what I thought. It's okay. So bacteria secretes toxins only when the population reaches a certain cell density, and then that they coordinate that expression through quorum sensing. Now, who here was like, what is this thing? I've never heard of quorum sensing. Maybe you have covered it in your content. Maybe this is a very first time. I think one key thing to note about in sciences is that when you encounter something you've never seen before, and I talked about this last week when we talked about like root words and how important they are, don't get stuck. Even if you've never seen this before, we can figure it out. We could like break it down and try to use context to figure out, what do we think the word means? So what do you think? What do you think quorum means? Maybe like in this time period, you're like, I don't know, I'm gonna go Google it. Don't quite yet. I want you to try to make an educated guess as to what you think quorum sensing might mean. And then let's see if that's verified with what the data says, what the context says, even with the word quorum itself as a definition word. What do you think that might mean? So go ahead and throw me your guesses. What do you think quorum sensing means? So yeah, it absolutely is, Beryl, and welcome back. It is a way of communicating between cells, hence this is why this is under the cell communication aspect. Quorum means like a gathering or a meeting, right? And it is based on density for well done. So you guys have tackled this somewhere in your content review. Excellent. Okay, so it's a way of coordinating stuff like gene regulation, a synchronicity that exists between a big grouping of cells or tissue or something like that. So that's exactly what it is. So if you know the content and you can understand what the hypothesis is saying, then the next step is to take a look at the data. So really important when you are analyzing data is like, here's my like key things. I do this at a glance now because I've had a lot of practice doing it. But when you're new to analyzing data, here are some key things to keep in mind. What are your independent variables? What are your dependent variables? And what are the trends? So independent variable, I can see is that it's time because generally speaking, the independent variable is on the x-axis. And then there's one more independent variable. And the key, if you don't know that you've got more than one independent variable, take a look. If there's a legend present, usually that's a second independent variable. So what do you think? Based on my nice little clue, what do you think would be actually in this case, yeah. No, there's a second independent variable, but I want you to tell me, what do you think? What are we also kind of controlling for or manipulating or something like that? If I take a look, I'm assessing also, in addition to sampling over time in the context of like the supernatant as well as the number of cells, okay? Actually, wait, let me think. Yeah, so the toxin, now it's not, maybe we'll hold back on that. Generally speaking, that's going to give you, yeah, so let's do this. Number of cells, and then the level, I guess, the concentration, this is my little like brackets, concentration of toxin. Nah, yeah, okay. Yeah, we're gonna just assess it over time. Yeah, so let's leave it at that. And then we'll stick with the independent variable just being a measure of time. Dependent variables are the assessment of the number of cells and the concentration of toxin. So tell me, what are your overall trends? And assess each one of these dependent variables separately with respect to time. So one would be over time, or as you increase time, right? What happens to one of my dependent variables? Over time, I can see that they both increase, but they have different shapes to their lines, and that's actually pretty important. So over time, what happens? So if I consider the cell part, I can see that it does increase, but it's like, and if you've done some like lab work, or you've done this part in your content review, it's almost like a logarithmic growth. Yeah, so as time increases, overall number of cells, so they are both a positive increase. Like if I was to draw arrows, they both do increase over time, but the shape of the line is actually kind of important as well. So this one plateaus up, right? Plateaus up, and then sharp increase right away. Yeah, I love the word that you used there, Nicole. So this is kind of like what I would call a log growth. It's a sharp increase initially, and then it kind of plateaus out fast. Whereas the second trend is over time, I see more of an exponential growth increase in the toxin. So this one is for the cells. Okay, this one's for the toxins. Nice, well done. Okay, so notice that the amount of toxins that is secreted or present is lower initially, and then it really goes up. Now, they're not comparable levels. Like, don't worry about that, because clearly the axes on the two y-axes here are going to be probably different units, but it's the shape of the line we want to pay attention to. Now, there is a part of content here that I want you to pay attention to. What is supernatant? What is that? Doesn't mention it in the passage whatsoever. So this is something that you want to understand about a lab technique, and that's going to be actually pretty key in devising the appropriate experiment to test the hypothesis. So I want to make sure that you guys all know, what is a supernatant? Say it exactly. Yeah, number of cells reach a threshold, and then you see the toxin, and that definitely is verified by the information in the stimulus. So nice job. They all kind of fit together. So what does the word supernatant mean? Supernatant. So I'll give you a hint. It has to do with a test tube. So if I've got a nice little test tube. Ah, okay. So I mean, the broth is what we generally use to say is the thing that we're culturing the bacteria in. And that's not wrong, Bronwyn. It's not wrong to call it what the bacteria is growing in, but supernatant versus the other thing, which is called the pellet. Okay, so when I take a suspension of cells, and then I have it in like a test tube like this, and then I spin it in a centrifuge, I get two parts. I get a solid pellet, and then I get the liquid that is surrounding it. Okay, so if I spin this, I get a big accumulation, a big clump of solid at the bottom, and then I get this thing at the top. So this is what we call our pellet. And then this is what we call our supernatant. Supernatant. Okay, so what do you think is in our pellet? What do you think is in our pellet? So if you understand how centrifugation works, right, it's essentially just spinning it, taking your test tube, you put it in this machine and it spins it in a circle really, really, really fast. And some, ultra centrifuges do it insanely fast. Oh, it's not quite the way speed, but I know where you're going with this, Bronwyn. Think about it. Is it that the heavy stuff or the light stuff are going to be in your pellet? It's not quite the ways, but I want you to think about size, really, to help understand the distinction between these two. What do you think? So supernatant is a liquid, solid is at the other end. I'm not quite telling you yet, but I really want you to work through this. What do you think? It could be waste, as Bronwyn mentioned, but what are these particles that Nick is probably referring to that's sitting in our pellet? It's probably not going to be a super, super fast, but it'll at least be the heavy stuff. So usually what is distinguishing between, what stays in the supernatant, what stays in the pellet is going to be size, okay? Yeah, exactly, Flora. The pellet will be heavier. The heavy stuff is in the pellet, right? And so if you think about this, we don't do anything. We're just taking a sample out of our suspension, and nice, Aditya, exactly, the cells are going to be at the bottom. So what's going to be in our supernatant, at least with respect to this particular stimulus? What's going to be in our supernatant? So I spin it out, all the cells accumulate at the bottom. I can separate it out from the supernatant, and what's going to be left behind in our supernatant that's important to the stimulus. There are many, many things that are true about the supernatant, but one thing I'm looking for in particular, and this is going to be really the key component for the experiment to test the researcher's hypothesis. So if the cells are sitting in the bottom, everything else, including, so here, let me just, where's this? There we go. So it's going to be size that differentiates between these two. Nice, well done. So the cells are going to be in the pellet, the toxin, and really everything else is going to be in our supernatant. Okay, so with this in mind, let's take a look. So that's enough information. That's the information you need in order to answer this question. Okay, so I'm going to give you a couple seconds, and I really want to see like what you are thinking with understanding how to test this hypothesis. With this understanding, go to take a couple seconds and type it out and tell me what you think would work to test the researcher's hypothesis. How would you prove, or at least support, that this is a type of cell communication called quorum sensing that's just mediated by that diffusible inducer molecule, this what we call an auto-inducer. So I'll give you a couple seconds. Go ahead and type it out and tell me, how would you devise an experiment based on the way that we analyze this graph and the data? Okay, so continue to type if you haven't already had a chance to completely enter in a proposal here. What I would do is I would take a sampling in two different places. I take a sampling of my cells. And maybe I'll just use a different color here. I take a sampling of my cells, like maybe this time point down here, early on, when there's an increase in amount of the cells that are growing, but low amounts of the toxin. That's probably in my supernatant. And then I take another sampling of my cells over here. right? So I've got high amounts of toxin as well as high amounts of my cells. Nice. Ah, okay. Okay. So if I want to prove that the toxin is mediating that that kind of quorum sensing, that it's essentially almost like a feed-forward reaction where, you know, the release of the toxin induces the other cells to release more toxin. If I took a sampling of these two cells, and then for the initial cells, I took a suspension. So I took the initial cells, so I'm going to call this like sample 1, and I'm going to call this sample 2. If I took the cells, and this would be the pellet, right, in sample 1, and then I resuspend pellet in sample 1 with what? What would I want to resuspend this in order to prove that the toxin is the thing that is the inducer molecule? What would I want to resuspend the cells in sample 1, which are just growing, but there's no toxin really around? What would I want to resuspend that pellet in? So Nguyen, like I like where you're going with that. You're right. If the pellet is smaller, there are less toxins, but it's not really quite proving that the toxin is mediating that initial kind of like big upward amount of growth and that signaling that exists between cells. Ah, see, I didn't think of that. Actually, that's a really good idea. Resuspend means essentially taking this clump of cells and then kind of mixing it back up, making it like, you know, making it kind of, it's not really dissolved, but I guess you can use that kind of word, dissolve it back up into more liquid again. So it's not quite water. It wouldn't be water. If I resuspend these guys in water, they'd probably explode. But it would kill the cells in any case. What I want to do is take that solid clump, that solid pellet, and then mix it back up so that it's like hanging out all free and loose with some kind of liquid. But which liquid should I use that? Which liquid should I use to essentially prove that there's quorum sensing that's existing in this particular system with C. difficile? So Nick gave a really good hint. Which supernatant, I guess, maybe that would be a better hint. Which supernatant should we be using to, you know, mix it back up, therefore be able to prove that the presence of the toxin is the diffusible inducer molecule? So if you're thinking that we need to resuspend pellet in the supernatant of sample 2, then you'd be correct, right? What is special about the supernatant sample 2? Well, that's chock full of toxins, right? And if you wanted to try to prove that the presence of the toxin is what's going to trigger all the other cells to release a bunch more toxins, then you would probably jumpstart that. Yeah, it would be the nutrient broth, but make sure you understand it's not just any nutrient broth, but it's going to be the supernatant of you know, of a lot of toxins, right? Nice, Nicole, exactly. So if you're like, I want that in word form, tell me how to like write this. Let me just show you what I mean. So this would be a sample response that would kind of fulfill the criteria. So you want to resuspend cells from early stages of growth, like sample 1, in the supernatant conditioned by cells in the late stage of growth, which is essentially what sample 2 says. So if the late stage secretes a signal in the molecule, in the medium, if the hypothesis is correct, it would then induce that quorum sensing and in cells that haven't reached a really high density yet. So notice the cells, the amount of cells I have in sample 1 is quite low compared to what I expected when it's reached its max growth. Okay. So it's a tough question. If you're like, I don't quite understand, you want to understand like what part you're not getting. So was it the technique? Was it the centrifugation in the words like supernatant and pellet? Was it the honing in on what the hypothesis was saying? Like, was it an issue with understanding the stimulus? Was it that you had a hard time kind of picturing this all in your head? or was it the aspects of the experimental technique itself? If you can understand what I wrote here, resuspending the cells, then I would say that like it's just a matter of practice and getting more exposure to this style of question and understanding like how experiments are designed in general. Yeah, Nicole, exactly. You're testing the pellet, the sample 1 cells with the conditions of the supernatant in sample 2. Yeah, so essentially I've got 2 sets of these test tubes, sample 1 and sample 2, and I'm kind of mixing matching between them to essentially prove that the toxin is responsible for that increase in growth. Okay, nice job, nice job. Okay, so this is like, again, we don't know exactly what the College Board is going to go with, you know, whether they're going to do 1 stimulus and like shorter sets, right? So less questions per stimulus, or whether they're just going to continue to do the big like free response, like the long form and then the short form. So I gave this as an example of a possible short form. What I'd like to do is give you a chance to tackle a long form free response question now. So again, I'm going to give you time to read through a part of the stimulus. The stimulus for this one is a fair bit longer here. So what I thought would be helpful would be to kind of break it up into chunks. And so what I'm going to do is I'm going to flip here. I'm going to flip here and clear this. And I want you to just take maybe about a minute to read through it. Now, there's no question here. So I just want to get you to get a chance to read it through. Take notes if you think it'd be helpful. And then I'm going to flip you to the next part of this question, and then give you a chance to tackle the question as well. Okay, so give yourself about a minute just to read this through. Take notes if you think it'd be helpful. We'll talk about it together, and then I'm going to give you a chance to take a look at the next part of this. Okay, so 1 minute for this part of the prompt. Okay, welcome back. Welcome back. So here we have a pathway. I will admit I've never heard of this pathway before. I thought it was pretty interesting. It's this alternate pathway that exists to metabolize glucose into pyruvate, which is very similar to how it works in glycolysis, but it's just like this side path here. And you don't really need to know much more about that. It's just giving you some history as to like the context for this particular enzyme, which is glucose 6-phosphate dehydrogenase. And then you're given the reaction for it. So glucose 6-phosphate is going to be turned into 6-phosphogluconate. And in the context of doing that, you've got your NADP plus and then NADPH. And so these are just like NAD plus and FADH. FAD plus, those are electron carriers. This is just a different kind of an electron carrier, and that is all catalyzed by this enzyme here. So this is the focus of this. And so it's very common to see that in stimuluses. They give you kind of like a context, like a pathway that is the background knowledge, but they don't really care too much about that. They're more considering, this individual enzyme or this individual step that's occurring in the pathway. Okay, so that's 1 minute here, not a whole lot of analysis. The real crux of this is going to come into the next part. So here I'm going to give you 3 minutes again. So 3 minutes to read the rest of the stimulus, which includes a fairly large table, and try to work on your first question. That's also on screen. And then what I'll do is afterwards, I'll give, we'll talk through the rest of the passage, as well as walk through the approach to the second question here. Okay, so 3 minutes to read the rest of the passage, and attempt that first question on your own. Okay, come on back. It's just like lab time. You've got some data and you're asked to graph the information. So the key components that you want to focus on when you are being asked to graph results. So this is one of the things that is often expected of those longer free response questions is making sure that things are labeled properly and that the scaling on the axes are appropriate. Now, they recognize that, yeah, oh, hey, Wendy, nice. Nice. Okay, why don't you all don't maybe not read Wendy's response, but like go ahead and tell me, how would you go ahead and graph this? I think that's excellent, actually. I really like that summary. Nice, Nicole. Yay, yay. That's excellent. So yeah, so if you agree or if that's exactly how you did it, feel free to chime in. If you're like, no, I did something totally different. I feel like a lot of these are fairly subject to how you understand it. I mean, there's always going to be a right or wrong. It's a standardized test, but there's a couple of different ways to approach this. And so Flora, if you did the same thing, awesome. Okay, so just real quick, you know, we've got two different kind of bacteria, two different bacteria, and they both express, I guess, this the glucose 6-phosphate dehydrogenase, and they're looking for a pH optimum. And then we're, you know, experimental setup, and we're told that we're going to detect the formation of NADPH, which is the product of this reaction, at a certain absorbance, and it's shown in the table. Bronwyn, well done, well done. Hopefully this was a fun exercise. I kind of like this actually. So here's what I would look for if I was like grading this. You want to write a line for each of the two axes, like that. And I think, you know, if you understand that you are changing the pH and you're measuring a response in the absorbance, then that will also just dictate exactly where these axes are supposed to be. So pH is going to be on the bottom because that is what we're changing. And so I would say this is 1, 3, 5, 7, 9. And so essentially I just skipped, you know, a box for every, so just one box per pH unit. So that's how I did my y-axis, on my x-axis, sorry. And on my y-axis, that is my, I'm going to call this change in absorbance at 365. So it's at 365 nanometers. And there's no like unit for this guy. Same thing with pH. So there's no units there. You don't need to add that in. And so it looks like I go all the way from like, you know, 0 to 1. I think that's an appropriate way. So this would be 0. I'm going to go in units of 0.2, very similar to how I did it with the pH. Right, so every other line I'm going to call this 1.0, like that, okay? And so we've got two different types of bacteria, and therefore, as Wendy mentioned, we should have two different kinds of bacteria, so two different lines. And so what I would recommend you do is just use different things. So like I'm going to call X is going to be bacteria A, and then I'm going to go O's for bacteria B, and so that will be my legend, right, for this graph. And so for your sake, I'm going to change the color. I know that's not an option for you when you're doing this on your own task, but just for visual sake, then that's what I'm going to do. So let's see. Bacteria A starts at a pH 1 at 0.22. So that's like roughly, these are my X's here, right, 0.85 here, 0.51 here. So remember, like I'm trying to like roughly guesstimate these, but I'm not like gonna stress myself over what looks like 0.06 and 0.08. So that's not something that you're expected to do, but you want to be kind of like roughly in the range of it, right? So something like that, okay? And so I would join the dots, like so. So you can see that there's a clear like peak to this thing. And then I'm gonna switch colors. For the other one, we'll do green, and then that will be my O's, right? So 1 will be 0.8, so something like that. And then 0.25, 0.54, got a bit of an overlap here. It's all good. 0.91, whoops, I did not mean to do that. I would say that the hard part of doing graphing is setting it up so that the axes make sense and that you've got the right X and Y axis. It's not necessarily like plotting this. Once you have this all set up, this should be relatively straightforward. And then 0.32, like that. And so clearly, you can see that they have different kind of optimums. They have different peaks, right? Maybe not, oh man, the race doesn't want to happen. Okay, so what I'm gonna do is I'm gonna X this out. Don't worry about this thing. It really shouldn't be expanding beyond the data that you're provided here. So yeah, that's it. This would be full points for a graphing question for a free response. Okay, so make sure that your axes are labeled and you've got a correct scaling, and then your data is correctly plotted with your pH as your independent variable, and you've got a nice little legend for both of those two types of bacteria. Okay. Well done. Okay, so let's move on. Let's look at part B for the same stimulus. Now, this one is asking, the student hypothesizes that bacteria A is an acidophile. Explain why this claim is supported by using the data. So here, I want to give you a chance to kind of work through this one on your own. Make sure you're not using gut feelings here. You want to provide some proof as to why you think the claim is supported. Oh, great question. So Flora and Farhan are asking, do you need a title? I took a look at a fair number of the graph style questions that are explained. I never saw in the oh, what's that called? In the description of like what they're using to grade these free responses, the requirement, yeah, the rubric is what I'm looking for. Yeah, the rubric doesn't require the title, but I would like to double check that. So I'm not 100% sure, but I never saw any rubric requiring a title. But if you felt like that would be a good thing and you had the time, I don't think that would be wrong to do so. So I think making a title just to be safe would be nice, but I've never seen any rubric require a title. And then in terms of whether you can make it a bar graph, well, it doesn't specify a graph, does it? So I think it would be very fair. If the question said bar graph, you got to stick with a bar graph, but it just said a graph and it didn't specify the kind, then I think doing a bar graph would be also completely fine. Nice questions, though. I really like that. Awesome. Okay, so I'm going to give you a minute to work on this next prompt on your own. Remember not to use feelings, but to use data to support why the student thinks that bacteria A is an acidophile. So I'll see you back in 1 minute. So go ahead and type in your answers as you kind of complete your line of reasoning here. Flora's got a really good one, but don't feel like just because Flora's done that and yours might be a little bit different. I would love to hear all sorts of different kinds of reasonings. Yeah, let's take a look at this. Tell me, what does an acidophile, acid, ophile, meaning phile means to do what? What does the suffix phile mean? It's the opposite of phobe. What do you think this phile means? These endings come very handy in like other realms of chemistry. Specifically, I think of like nucleophile, acidophile. There's other like suffixes that rely on this kind of understanding. Flora's got a pretty good one there, but go ahead and tell me, what do you think this phile means? What does the word phile? So if you're, I actually don't know, is it Latin or is it Greek? Huh. Yeah, well done. It is attracted to Rashi, exactly. So this is like a big old heart. They love acidic conditions. They love acidic conditions. So if you're kind of thinking about this, well, the work that we did in the first part of the question actually is a really big helping hand, because if you didn't notice the optimal pH in which these bacteria thrive in, one is clearly a different optimum to the other. So exactly, so it is a low pH. So in terms of content, you had to know that an acidic pH is from the realm of less than 7. So that would be an acidic pH. Phile means loving. Yeah, well done, team. Well done. And so because its pH optimum is roughly around 3, according to the data that were given in this graph, that would be the support for why this would be an acidophile. So if you're looking for a text version of a correct response here, it would be something like this. An acidophile is a bacteria that thrives under acidic pH conditions. And notice that we define acidic pH. You're not just assuming that this is knowledge. This is something that you do want to explained. So it is defined as less than pH of 7. According to the data, the pH optimum of the bacteria is around 3, or you could have specified it as greater than 1, less than 5, which is an acidic pH. I love it. Nice. Is it Greek? Flora, see, this is how much I don't know. You guys are all teaching me. Well done. Okay, so we got one last part to this question, and I do like where you guys are all going with this. So actually, gut check before we take a look at the last part of this question. I think they do test different skills. The multiple choice questions tend to be a lot more content heavy. The free response questions tend to be a lot more analysis and scientific reasoning kind of skills. Which are the ones that you find more comfortable with? You know, is it that you need more content work? Then definitely go ahead and do that. And I think that doing more multiple choice questions, even though they're not specifically tested, is a great way to ramp up that content. But if you're like, I get the content, I'm fine with all the content, it's just all this like analysis stuff that's hard, then your task, you know, to practice for your upcoming test is to make sure you seek out more free response questions. And so it's 2 different methods of studying depending on what you need for the future. Okay. Okay, so one last question to go. I'm just going to clear this. And so again, let's take about a minute to work through this next prompt on your own, and then we'll talk about it together. So there we go. Last one, and then we'll talk it over. So remember, there's 2 parts to this. Make sure you propose as well as describe, and so your complete answer should fulfill both of those conditions. 1 minute for this question. Okay, team. So this one is asking about you to propose a control treatment and then just describe why that would help support the information that you're getting from the results. So before we propose some control experiments, and because that is an open-ended question, there's multiple things that you can propose and support that would be totally fine and would get you the points. But before we do that, tell me, what is a treatment? I mean, what is a control? What is a control? So let's think. Well, controls are often given as a way to verify whether something is going to give you the positive effect or the negative effect. So those are the 2 types of controls. You can have a negative control or a positive control. So if in this case, you know, you're assessing the pH optimum and then you're looking for a change, well, one way to do this would be to consider like doing the thing. Yeah, Wendy, that's one way. And if you follow that up, if you do it without the enzyme, that would be a negative control, because if you take away the enzyme, you're not going to get the actual change in the result. There are other ways of doing this as well, and Rashi mentions different conditions, but remember, that wouldn't give you the points. You need to propose what kind of different conditions and support why that would be then a control. So one of them, and I'll go ahead, kind of like a baseline level, but remember to be specific here. Yeah, be specific. If you're proposing something, here's one. I'm just going to go ahead and type it then. Yeah, so if you did the reaction without enzyme, which would then support or describe or confirm that the enzyme is necessary, that would be one type of control. So reaction without enzyme to confirm that the enzyme is necessary for this reaction to occur, that would be one type of control. And to be clear, this would be, sorry, one second. This would be a negative control. So negative control. So Rashi says, control the incubation with G6PD and determine the amount of NADPH and how it changes. So would that fulfill the criteria in this one? We're not really assessing whether or not the enzyme works. We're assessing the pH optimum here. And so while that would be an experiment that you could do, Rashi, I'm not entirely sure how that fulfills the criteria of a positive or a negative control. Yeah. And so, and I apologize if I mispronounce anyone's name. And I recognize that a lot of them are handles and not actual names, but Rugoax. Yeah, you're right. The control has to prove something that you're not testing. It's not affecting the experiment itself. Yeah, so it's not, okay, so here, if you take away the enzyme and the enzyme is required for this to happen, then you can assess whether or not the enzyme is necessary for this reaction to occur. Another negative control would be to take away another part of this. So maybe if we took away the other part of the substrate, so like glucose phosphate or NAD+, would all assess as a part of a negative control. Yeah, so all it is to help you understand what if the experiment is working the way it should. Another way to do a negative control would be to do this. Maybe, let's say it's not just the enzyme activity, but maybe it's just the presence of the enzyme. Maybe it's like a binding protein of some sort. So a way to assess whether the active enzyme is necessary would be then to have the reaction with the denatured enzyme. So if you heated up that enzyme to really, really hot and denatured it and still added in the correct amount, then it confirms that you need an active enzyme in order for this reaction to occur. So it'd be another type of a negative control. A final control that I thought of, because I was like, oh, these are all negative controls. I really want to see an example of a positive one. So what if we pulled it from a confirmed acidophile? So we knew the bacteria, it wasn't an unknown bacteria. And then we did that same reaction under a G6PD from an actual acidophile to confirm the optimal pH. And if it had the same kind of optimal pH, that would further support whether or not bacteria A is an acidophile. Okay, so the first two, let's see, the first two are negative controls, right? And this one is a positive control. Positive control meaning it gives you the result that you're looking for because you know it has to occur because we know the source of this bacteria, okay? So hopefully this was a great review for you. I do encourage you to continue to practice. These are some tough skills, some tough questions that we've gone through, but they are certainly doable just the same way that content is learned. If you come back tomorrow, we're going to do something very similar, but we're going to do in the context of metabolism. So we'll do some photosynthesis, anaerobic and aerobic respiration. But other than that, you know, I hope you guys all have a great afternoon, and I do hope to see you again soon in a future session. Thanks as always to the fantastic off-camera instructor, Nick, and you know, see you back tomorrow.